Mites are among the most overlooked organisms on Earth, yet they represent one of the most successful evolutionary groups ever to exist. As members of the subclass Acari, they are closely related to spiders and ticks, but unlike their larger relatives, most mites exist entirely beyond human perception. They inhabit almost every environment imaginable, from deep soil layers and forest canopies to freshwater systems and even extreme habitats. In a single handful of soil, there can be thousands of individual mites, each playing a role in maintaining ecological balance. What makes mites particularly fascinating is not just their abundance, but their diversity. Under magnification, their bodies reveal intricate structures, specialized adaptations, and in some cases, extraordinary coloration. While many mites appear dull or translucent, a select few display vivid colors, complex surface patterns, or even optical effects that rival those found in much larger animals. Among this vast diversity, certain species stand out not only for their biological function, but for their visual impact. These mites demonstrate that even at microscopic scales, evolution can produce forms that are both functional and striking. The red velvet mite is one of the most immediately recognizable mites due to its intense coloration and distinctive surface texture. Unlike most mites, which remain hidden and unnoticed, this species often becomes visible after rainfall when it emerges onto the soil surface. Its body is covered in dense, fine setae, creating a velvety texture that gives the mite its name. The surface is not merely aesthetic, it may help retain moisture and protect against environmental stress. The bright red coloration is produced by pigmented compounds and serves a critical defensive function. This is a classic example of aposematism, where a bold color signals to predators that the organism may be toxic or unpalatable. This warning is not empty. Red velvet mites are known to produce chemical defenses that deter predation, making their coloration an effective survival strategy. Their life cycle adds another layer of complexity. In the larval stage, these mites are parasitic, attaching themselves to insects or other arthropods and feeding on their bodily fluids. As they develop into nymphs and adults, they transition into free-living predators. At this stage, they actively hunt small invertebrates and eggs, contributing to the regulation of arthropod populations in their environment. This combination of vivid appearance, chemical defense, and dual ecological role makes the red velvet mite one of the most remarkable and visually striking members of the mite world. The genus Tuckerella presents a very different type of beauty, one based not on bold color, but on structure and symmetry. These mites are often referred to as peacock mites due to the distinctive arrangement of their posterior setae. Extending from the rear of the body are long, filament-like structures arranged in a fan-shaped pattern. These setae are precisely organized, creating a balanced and almost ornamental appearance. Under magnification, the arrangement appears highly deliberate, with each filament contributing to a symmetrical display. Functionally, these structures serve as sensory organs. In a microscopic environment where visual cues are limited, the ability to detect vibrations, air movement, and physical disturbances is essential. The elongated setae allow the mite to interpret its surroundings with high sensitivity. Tuckerella species are typically found on woody plants, including bark and stems. They feed by piercing plant cells and extracting their contents. Despite this feeding behavior, they are generally not considered harmful at a large scale, as their populations tend to remain low. What makes them particularly fascinating is how their functional anatomy results in such a visually striking form. The combination of symmetry, extension, and fine structural detail creates an appearance that is both precise and elegant. demonstrating how biological necessity can produce unexpected visual complexity. The genus Rainbowia introduces a completely different mechanism of visual expression. Unlike the previous mites, whose colors are produced by pigments, these mites rely on structural coloration to generate their appearance. Their cuticle contains microscopic features that interact with light in highly specific ways. Through processes such as interference and diffraction, these structures selectively reflect certain wavelengths of light, producing colors that change depending on the angle of observation. As a result, Rainbowia mites can display a shifting spectrum of colors, including blues, greens, purples, and metallic tones. This iridescent effect is not static. It changes dynamically as the mite moves or as the viewing angle shifts. This type of coloration is also found in other organisms, such as certain insects and birds, but its presence in mites highlights the complexity that can exist even at microscopic scales. The precision required to produce such optical effects suggests a highly specialized structural arrangement at the nanoscale. Although their ecological roles are less extensively studied, they are believed to participate in micropredation, or the processing of organic material. Similar to many other mites, their primary significance lies in their unique optical properties, which have attracted interest in scientific research related to biomimicry and materials science. Members of the family Erythracaridae represent another group of visually striking mites, characterized primarily by their bright red to orange coloration. At first glance, they may resemble red velvet mites, but closer examination reveals important differences in both structure and ecological role. Their bodies are typically smoother and less densely covered in setae, giving them a more streamlined appearance. The coloration is again derived from carotenoid pigments, which play multiple roles in protecting the organism. These pigments can help shield against ultraviolet radiation and reduce oxidative damage, both of which are significant challenges at small scales. These mites are most commonly found in soil and leaf litter, where they are closely associated with decomposing organic material. In these environments, they contribute to the breakdown of organic matter, playing a key role in nutrient cycling. By processing decaying material, they help convert it into forms that can be reused by plants and microorganisms. Their presence is often an indicator of a functioning and balanced ecosystem. Although they may not exhibit the elaborate structures seen in other mites, their coloration and ecological importance make them a significant and visually notable component of the microfauna. Conoppia palmicincta represents a different form of visual complexity, one based on structure rather than color. As an Oribatid mite, it belongs to a group known for its hardened exoskeleton and intricate body design. The body of this mite is composed of clearly defined plates and ridges, forming a highly organized and symmetrical structure. The surface is often sculpted with fine details, giving it an appearance that resembles a miniature piece of carved material. This structural complexity serves practical purposes. The hardened cuticle provides protection against predators and environmental stress, while the overall body design contributes to durability and longevity. Oribatid mites are known for their relatively long life cycles compared to other mites, and their robust structure is a key factor in this resilience. Ecologically, Conoppia palmicincta plays an important role as a decomposer. It feeds on fungi, algae, and decaying organic matter, contributing to the breakdown and recycling of nutrients within the soil. This process is essential for maintaining soil fertility and supporting plant growth. The visual appeal of this species lies in its precision and detail.